Dear students, hello and assalamu alaikum. I am Ali Abbas Qazilbash and I would like to welcome you back to our program and this discussion on the topic of photosynthesis. I'm sure that you all know the basic definition of photosynthesis. Photosynthesis is the process by which green plants and some bacteria use the energy from sunlight to convert carbon dioxide and water into sugar and oxygen. The sugar is then used in various metabolic activities to produce adenosine triphosphate, ATP, the chemical fuel used by all living things. Here, I would like to add that photosynthesis, which is the conversion of unusable sunlight energy into usable chemical energy, is associated with the action of the green pigment chlorophyll. Now, the overall reaction of photosynthesis is basically 6 molecules of carbon dioxide plus 6 molecules of water will give you 1 molecule of the sugar glucose plus 6 molecules of oxygen. Green plants, in the presence of sunlight, use 6 molecules of carbon dioxide plus 6 molecules of water to produce 1 molecule of sugar plus 6 molecules of oxygen. Now, these sugars, or carbohydrates as they're better known as, are then broken down in the mitochondria of the cell to release adenosine triphosphate, ATP. Now, students, this illustration shows the conversion from glucose to ATP. It is, in fact, a drawing to show the steps of aerobic cellular reaction, which will be explained in detail in our program of respiration. Plants, especially green plants, dominate our environment, and we should be very grateful to these plants for various services they perform for us and all living things. The primary function that green plants perform is the fixing of carbon dioxide in the presence of sunlight, the process of photosynthesis, during which they use sunlight, photo, as the source of energy to synthesize food, and also produce oxygen as a byproduct of this process. Therefore, it is without doubt that photosynthesis is absolutely essential for the continuance of life because it is the source of food, both directly and indirectly, for herbivores and carnivores respectively, and oxygen for all living things. With our discussion so far, we have come to know the importance of sunlight, the radiant energy. We also know that the food produced through photosynthesis eventually becomes the food for the rest of the world. For example, humans and all other animals either eat plants directly or eat animals that have eaten plants. In other words, we can say that the living organism that produce their own food are known as autotrophs, and those organisms that do not produce their food but depend on others for their food are known as heterotrophs. Therefore, when analyzing the food chain, we can say that autotrophs are represented by green plants, algae, and a few types of bacteria, and herbivores are those animals that feed directly on plants or plant products such as cows, goats, and rabbits, whereas carnivores are those animals like snakes, lions, dogs, and cats that feed on herbivores or other carnivores. And omnivores are those animals which feed on herbivores, carnivores, plants, or plant products like humans and bears, etc. Now let us discuss the process of photosynthesis in some detail, starting with how the raw materials are supplied and used, and where the process of photosynthesis actually takes place, the chloroplast, its structure and role. Now, plants get their water, the source of hydrogen, from the soil and absorb it through their roots. Carbon dioxide reaches to the inside of the leaf, the site of photosynthesis, from the atmosphere. Carbon dioxide enters the plants through pores called stomata, present on the surface of the leaf. The opening and closing of stomata depends upon the presence and absence of light and is powered through the process of osmoregulation, the absorption of water by guard cells surrounding the stoma. The source of energy used to drive this process is, of course, radiation, sunlight, that is incident on the leaves and absorbed by specific pigments present in the chloroplast. This diagram very clearly shows a leaf and its anatomy. The mesophyll cell, with its detailed internal structure, the granum, which are stacked with thylakoids, the site where sunlight is trapped and the process of photosynthesis is initiated. At this stage, my friends, you will certainly not be surprised to know that radiation energy from the sunlight or otherwise is part of the electromagnetic spectrum, which consists of various types of radiant energies, from the shortest wavelength gamma rays to the longest wavelength radio waves. The shorter the wavelength, the greater the energy. In this diagram, you can see the different types of radiant energies. White or visible light is only a small portion of the electromagnetic spectrum, which has various wavelengths of light. In this illustration, here you can see that only violet, blue, and red lights are better absorbed by both chlorophyll A and chlorophyll B, the pigments associated with photosynthesis. Let us now discuss the structure and function of chloroplasts. The chloroplast is the site of photosynthesis. It is an organelle found in the cells of green plants and certain algae. In the chloroplast, a double membrane surrounds a large central space called the stroma. A membrane system within the stroma forms the grana, so called because it looks like piles of seeds, and early scientists, the word grana meant seeds. Now, electron microscopy later revealed that each granum is actually a stack of flattened sacs or discs called thylakoids. The grana are connected to one another by stroma lamellae. Here, you should keep in mind that chlorophyll and the yellow-orange pigments, which are called carotenoids, are found within the membrane of the thylakoids, and these pigments are able to absorb the energy of sun. The space between the chloroplasts is called a stroma, which consists of an enzyme-rich solution. These enzymes incorporate carbon dioxide into organic molecules. Therefore, we can say that carbohydrates produced by photosynthesis are formed within the stroma. Now, there are two distinct phases of photosynthesis. The first set of reactions, which takes place in the thylakoid, is called the light-dependent reactions, or simply light reactions. The second set of reactions, which takes place in the stroma, are called light-independent reactions, or dark reactions, because they do not utilize solar energy. Now we shall discuss the light-dependent reactions in some detail. The components of a photosystem are several hundred chlorophylls and carotenoid molecules, plus the reaction center chlorophyll A molecule. Now, light energy is absorbed in the form of photons and then passed from molecule to molecules, as illustrated here in yellow, until it reaches the reaction center chlorophyll A molecule. Thereafter, energized electrons are passed to an acceptor molecule. During this process, the chlorophyll is activated by a higher energy, so that its two electrons move to a higher energy state and are captured by an electron acceptor called plastoquinone. The chlorophyll replaces its lost electrons by disrupting a water molecule, and this disruption causes the water molecules to break down into hydrogen and oxygen, a process called photolysis of water. This entire process takes place within the system designated as photochemical system 2. In photochemical system 2, from the electron acceptor plastoquinone, the electrons are passed through an electron transport system. This chain consists of a series of electron acceptors, which are cytochrome B, cytochrome F, and plastocyanine. Each electron acceptor is at a lower energy level than its predecessor. This way, the electron goes on losing their energy. Some of this energy is used to make ATP from ADP between cytochrome B and F molecules. In this way, ATP synthesis is generated by the light energy and is called photophosphorylation. Similarly, electrons are passed along another pigment system at the bottom of photochemical system 1, where light energy again boosts an electron to another electron acceptor called ferredoxin-reducing substance, as you can see in this diagram. From this acceptor, the electrons are passed along to a nucleotide of adenine called NADP. It is in its oxidized form and then is reduced to NADPH. The next important set of reactions are the light-independent or dark reactions. At this stage of photosynthesis, the NADPH and ATP molecules produced as a result of light reaction are used in the reduction of carbon dioxide. The reduction of carbon dioxide occurs in the stroma of the chloroplast by means of a series of reactions known as the Calvin cycle. The series of reactions involved in the Calvin cycle include carbon dioxide fixation, carbon dioxide reduction, regeneration of ribulose bisphosphate, RUBP. The diagram illustrates all three steps. Here you can see the Calvin cycle in its simplified form. You can see that ribulose bisphosphate, which in this diagram is written as RUBP, combines with carbon dioxide, forming a 6-carbon molecule, which immediately breaks down into two 3-carbon molecules, phosphoglycerate, PGA. PGA is then reduced to phosphoglyceraldehyde, PGAL. The NADPH and ATP are used in the reduction of phosphoglycerate to a triose or 3-carbon compound termed as phosphoglyceraldehyde. This is the end product of the cycle. Outside the cycle, the two PGA molecules can combine to give glucose 6-phosphate. For every 3 turns of this cycle, 5 molecules of PGAL are used to reform 3 molecules of RUBP. Now we know that the food does not just mean glucose or carbohydrates. There are other types of food as well, like proteins, lipids, etc. And you can see that there is a net gain of 1 PGAL molecule in the Calvin cycle. And this PGAL molecule is used to produce glucose and numerous other organic molecules. From these PGAL molecules, starch, cellulose, other carbohydrates, proteins, glycerol, fatty acid molecules are produced as seen here. Let us now summarize the process of photosynthesis with the help of a few diagrams. Photosynthesis basically occurs in the chloroplasts, which are specialized organelles present in all green plants and some algae. They contain stacks of thylakoids within the grana, where the light reaction of photosynthesis occurs. In other words, solar energy is trapped by specific pigments in these thylakoids, and the water molecule is broken down into hydrogen and oxygen. This process is part of photochemical system 2. The solar radiation arrives at the reaction center where the chlorophyll A molecule absorbs the energy and releases an electron, which in turn is passed on to an electron acceptor known as plastoquinone. From here, the electron is then passed along an electron transport system from one acceptor to the next, each having a lower energy state than its predecessor. This results in the release of energy, some of which is utilized to form ATP from ADP, photophosphorylation. Simultaneously, the water molecule breaks up into hydrogen, used in the reduction of carbon dioxide, and oxygen, which is released as a byproduct. Similarly, electrons are also passed along another pigment system called photochemical system 1, where light energy again boosts an electron from the reaction center to a ferredoxin-reducing substance, which subsequently releases it to a nucleotide of adenine, NADP, which in turn is reduced to NADPH, which is used as the source of energy along with ATP to drive the dark reaction. Chloroplasts also have a large central space called the stroma. It is here in the stroma where the next step of photosynthesis occurs, the dark reaction. During this process, energy from the sunlight is not utilized. Instead, energy from ATP and NADPH molecules is used. In the dark reaction, carbon dioxide is fixed through the Calvin cycle to form organic molecules such as phosphoglyceraldehyde, which is then used to manufacture organic molecules used as food source. During the Calvin cycle, carbon dioxide is fixed through the reduction by combination with a 5-carbon molecule known as RUBP, ribulose bisphosphate. Upon combination with carbon dioxide, RUBP forms a 6-carbon molecule, which immediately breaks down into 2 3-carbon molecules called phosphoglycerate, which are then reduced to phosphoglyceraldehyde, PGAL. From these PGAL molecules, starch, cellulose, proteins, fatty acid molecules, etc. are produced. To conclude, dear students, we can say that photosynthesis is a 2-phase process by which plants manufacture their own food. The first phase consists of the light reaction, which takes place in the thylakoids, where energy in the form of sunlight is trapped by specific pigments, and some of this energy is used in the breakdown of a water molecule into hydrogen and oxygen, which is released, and the remaining energy is used in the conversion to chemical energy in the form of ATP and NADPH. The second phase takes place in the stroma and is known as the dark reaction, where this chemical energy is used to fix and reduce carbon dioxide to form a 3-carbon molecule, PGAL, which is then used to make sugar and other food substances. With this, dear students, we've come to the end of today's program. I would like to thank you all for your undivided attention. Until we meet again, wish you the very best. Khuda Hafiz.
body is very complex. It consists of different systems. Each system is performing specific functions. For example, the digestive system digests the food we eat and makes it available for the body to use it for various functions. Kidneys excrete waste products from the body. Dear students, our today's program is about the structure and composition of blood and circulation of the blood in the heart. All the functions are performed at specific locations in the body. The materials needed for these functions, or those produced as a result of metabolism, need to be transported from one part of the body to the other. From your previous knowledge, you must be knowing that blood is the main transport system of the body. It carries digested food from the intestines to various parts of the body, waste materials, for example, urea and carbon dioxide, to the sites where they are excreted from the body, secretions from the glands to different parts of the body, oxygen from the lungs to each cell of the body. In order to carry such a variety of materials, the blood needs to have a special physical and chemical structure. To understand the functions of blood, we must study the structure and composition of blood. Here I have some fresh blood in this test tube. Look at it carefully. As you can see, it is a red fluid of comparatively thick consistency. You can see it is a viscous fluid and seems to have no particles in it. Let us see if it has any suspended particles in it. We will leave this test tube for a while, and if there will be any heavy suspended particles, they will settle down. Now, can you see any difference between these two test tubes? In this test tube, the blood has formed layers. The upper clear straw-colored portion is plasma, while the lower colored portion has the solid components, that is, the blood cells. To check it, if the blood has cells in it, we will examine it under the microscope. For this purpose, let us prepare a slide of the blood. This is a blood slide. Can you recognize the cells in this slide? There are three types of cells in this slide. These are red blood cells, these are white blood cells, and these are the third variety of cells, which is called the platelets. Now this card shows you the composition of the blood. Here is the plasma, which is a viscous fluid, and second is the blood cells, which I have already shown you. Now let's study the structure of plasma and blood cells in detail. First of all is the plasma. Plasma is the fluid portion of the blood. It is a dilute salt solution. An analysis of it shows that about 90% of it is water, which acts as solvent. Salts, which include sodium, potassium, calcium, magnesium, chloride, and bicarbonates. Their main function is to maintain the osmotic balance, pH buffering, regulation of membrane potentials. Third main component is the plasma protein, which are of three types: albumin, fibrinogen, and immunoglobulins. Their function is to maintain the osmotic balance as well as for pH buffering, clotting, and immune responses. Fourth component are those substances which are transported by it. Nutrients, for example glucose and vitamins, waste products of metabolism, respiratory gases like oxygen and carbon dioxide, hormones, and heat. 45% of the blood volume is due to blood cells. There are three principal types of blood cells, namely erythrocytes, leukocytes, and thrombocytes. We will study them one by one. First, the erythrocytes. If you look at their shapes carefully, you will notice that these circular, disc-like cells are the most numerous of all cells. These are red blood cells or erythrocytes. There are about 20 trillion RBCs in the human body, or about 5 million per cubic millimeter of the blood. Average life of RBCs is 120 days. In embryonic stage, RBCs are formed in the liver and spleen, but after birth they are formed in bone marrow of certain bones like ribs and breastbones and at the extremities of long bones. When RBCs are formed, they contain a nucleus. However, soon after their maturation, nucleus disappears. If we look at these cells, they are like a ball which has a depression on two sides. That is, they are biconcave cells. These are very flexible and can be squeezed through hair-like capillaries. Erythrocytes are full of a protein, hemoglobin, which carries oxygen, and red color of the blood is due to this protein. If you study the structure of this protein, you will see that each molecule of hemoglobin are 4 atoms of iron. Oxygen forms a bond with iron atoms where there is a higher concentration of oxygen, and hemoglobin is converted to an unstable compound which is known as oxyhemoglobin. Wherever in the body oxygen is needed, this bond breaks and oxygen is liberated. Old and worn out RBCs are destroyed in the liver and spleen. When RBCs are destroyed, the hemoglobin is converted into bile pigment and iron is used up for the formation of new RBCs. A deficiency of RBCs causes anemia, which results in the deficiency of oxygen supply to various parts of the body. And it also causes many other health problems. On the screen, you are watching a blood slide. Besides RBCs, you can see a number of other cells. These are leukocytes or white blood cells. They are less in number than RBCs, only about 7,000 to 11,000 per cubic millimeter of blood. WBCs and RBCs are in ratio of 1:600, that is, for 1 WBC, there are 600 RBCs. WBCs are larger than RBCs and do not contain hemoglobin. There are several kinds of WBCs, each with a different function. However, all WBCs work for the defense of the body against diseases. They are not confined to blood vessels, but they also migrate out into the interstitial fluid. There are two major types of leukocytes. These are the granulocytes and the agranulocytes. Granulocytes are very flexible, nucleated cells of irregular shape with a fine granular cytoplasm. They are produced in the bone marrow and they are of three types. Eosinophils, which represent about 1.5% of WBCs, but their population increases in persons with allergic conditions like asthma and hay fever. Their granules stain red with acetic or eosin dye. Basophils are about 0.5% of the WBCs. They produce two important substances, which are histamine and heparin. Their granules stain blue with the basic dyes such as methylene blue. Third type of the granulocytes is the most important, and it is the neutrophils. These constitute about 60 to 70% of the total WBC count. They are actively phagocytic and protect the body against invading microorganisms. They engulf and digest the invading pathogens. Now, the agranulocytes. There are two types of agranulocytes. These are the lymphocytes and the monocytes. Lymphocytes are about 20 to 30% of the WBCs. These are formed in the lymphatic system and spleen from the precursor cells which originate from the bone marrow. They have a large nucleus which is round or oval in shape and only a small quantity of cytoplasm. Their major function is to mediate immune reaction such as antibody production, graft rejection, and tumor cell killing. Monocytes have globular nucleus. Dear students, now the third variety of blood cells, that is the platelets or thrombocytes. Blood platelets or thrombocytes are a very important part of blood. They play a central role in blood clotting. Now let's see how the platelets stop the loss of blood from a ruptured blood vessel. Platelets gather where a blood vessel ruptures and blood cells are coming out of it. The first platelets to arrive form a plug across the opening. They release chemicals that convert the liquid blood protein fibrinogen to insoluble fibrin. Fibrin forms a mesh of needle-like fibers that trap platelets and other blood cells, creating a clot. Dear students, now here is Dr. Zishan who will tell you about the structure of heart.
We will tell you about the structure of the heart. This is a lamb's heart. Lamb is a mammal. The structure of its heart is quite similar to the human heart. If you touch it, you can easily feel that it is quite strong. It is strong because it is composed of special kind of muscle fibers known as myocardial muscles. Now, if you look, you can clearly see different types of vessels arising from the heart. This is the pulmonary trunk, this is the aorta, and this is the superior vena cava. And this is the atrium, and this is the ventricle. Now, for better understanding, we will try to teach you on a model of the human heart. This is model of a human heart. From front, the major vessels can be easily seen. This is aorta, this is superior vena cava, and this is pulmonary trunk. Behind the major vessels lies the trachea, and behind the trachea lies the esophagus. Now, to understand the circulation and the internal structure of the heart, we can open this model. Now we are looking through the right atrium. This is the superior vena cava and this is the inferior vena cava. The superior vena cava carries blood from the head and neck region and the upper limb, and the inferior vena cava carries blood towards the heart from the abdomen, thorax, and the lower limbs. The function of the valve is to allow the blood to flow only in one direction, that is through the right atrium into the right ventricle. The valve prevents the backward flow of the blood. The valve is attached to the walls of the ventricle through the special muscle fibers known as chordae tendineae. The function of chordae tendineae is to prevent the overstretching of the valve during the ventricular contraction. From the right ventricle, the blood passes to the lungs. In the lungs, the carbon dioxide is washed out from the blood and oxygen enters into the bloodstream. Now, from the lungs, blood comes back again to the left atrium. From the left atrium, blood passes into the left ventricle again through a valve. This valve is known as mitral valve. This valve consists of two cusps. This valve also is connected to the ventricular valves through chordae tendineae. Again, the function of the valve is to prevent backflow of the blood. We can see through this red wire the direction of flow. From the left ventricle, blood passes to whole of the body through the aorta and its branches. Now, to understand the circulation of blood through the heart and the body, we have a simple diagram for you. In this diagram, the blue color shows the deoxygenated blood and the red color shows the oxygenated blood. From whole of the body, the deoxygenated blood comes into the right atrium. Through the right atrium, it goes to the right ventricle. And from the right ventricle, it goes to the lungs. In the lungs, as I mentioned before, the carbon dioxide is washed out and oxygen enters into the bloodstream, so the blood becomes oxygenated. And this oxygenated blood passes to the left atrium. From the left atrium, blood passes to the left ventricle, and from the left ventricle, the blood passes to whole of the body through the aorta. From the aorta, blood goes to the stomach, intestines, kidneys, head and neck.
Dear students, now let's discuss about the vascular system. It consists of 3 different types of vessels: arteries, veins, and capillaries. If we cut a cross section of an artery like this, we can see the structure of blood vessels. As you can see, the arterial wall has 3 different layers. This outer layer is made up of fibrous connective tissue. Now look at the middle layer. It is made of smooth muscles and elastic tissues, while the innermost layer consists of an elastic membrane called endothelium. All arteries except the pulmonary artery carry oxygenated blood from heart to other parts of the body. Arteries vary in thickness. For example, the aorta is a very large artery, while there are small arteries too, which are about 0.2 millimeter in diameter. These are named as arterioles. Veins carry blood from the body back to the heart. In the veins, the blood pressure is comparatively less than the arteries. Their walls are not so thick as are of arteries. Although they are composed of the same 3 layers as are of arteries, but they are not very thick. Particularly, the middle layer is much thinner and not so developed as it is in the arteries. The innermost layer of endothelium is absent in some smaller veins. Here, there is a vein. If we insert this pencil into it, you will feel that we can do it easily, only in one direction. It is difficult to insert the pencil in the opposite direction. It is so because there is valve which opens in only one direction. All the veins have valves which prevent blood from flowing in backward direction, thus keeping the blood flowing towards the heart. A third type of vessel, named capillaries, form a network in the tissues. They are of microscopic size and have very thin walls through which materials can be exchanged. Dear students, now let's summarize the program. The composition of blood. Blood is composed of a fluid part, plasma, and a solid part, blood cells. Blood plasma is composed of water and metabolites, waste materials, and salt ions. Blood cells are of 3 major types: erythrocytes, leukocytes, and thrombocytes. Now, the structure of heart. Heart is a muscular organ. Human heart is divided into 4 chambers, 2 atria and 2 ventricles. Right and left side of heart are separate while upper and lower chambers are connected. Dear students, our today's program was about the structure and composition of blood as well as on the circulation of blood in the heart. See you next program. Allah Hafiz.